Check if you have upcoming already on Omega Bio-Edutainment.
Lo-fi ambient with soft piano synths and gentle lab-like sounds. Perfect background of focused hands on practicals. Lo-fi ambient with soft piano synths and gentle lab-like sounds. Perfect background of focused hands on practicals. Lo-fi ambient with soft piano synths and gentle lab-like sounds. Perfect background of focused hands on practicals. Lo-fi ambient with soft piano synths and gentle lab-like sounds. Perfect background of focused hands on practicals. Lo-fi ambient with soft piano synths and gentle lab-like sounds. I'll be electronic track, synthesis and digital.
Bioinformatics practicals for today is going to be protein structures and functions. This is actually specifically made for microbiology students, and the tools we are going to learn how to use today is going to be for protein structure determination, as well as the tools that will be available for determination of their functions. This includes UniProt, PDB, Expasy, and PyMOL. Also, in this presentation today, we are going to see a lot of demos, that is demonstration, exercises, and what are the offline tips that you should know as a student of microbiology. Just like I said, determination of protein structures and function. Now, what are the roles and structure of protein? Protein perform essential functions in microorganisms because definitely all enzymes are proteinous in nature, but not all proteins that are enzymes. So that has to be established. Then the structure will actually determine the function, as I normally say, because the structure is where we are going to have the active sites, and the way protein is being folded determine and go a long way to determining its functionality. So as you have it on the screen, you will see that inside a typical enzyme, inside a typical bacterial cell, you have the enzyme and you have the transporter, that is transporters of a protein. That is, whenever you have any protein, protein will perform specifically any of these three functions, or it can perform all the three. As an enzyme, it can act as a transporter that is going to transport materials from one point to another. And sometimes we also have some proteins that are structural, that is, they contribute to the structure of the microorganism. An example of what we have here is legemoglobin, which bind at a specific site, making use of oxygen. What are the levels of protein structures? Basically, there are four levels of protein structures, which are the primary level, that is the sequences of amino acid. So if you retrieve, you go to NCBI and you retrieve amino acid, so all those ones are at their primary functions. Then the next one is which is secondary functions, structures, which can either be at alpha helix or beta helix. So the third one is the 3D folding system, which is the tertiary. This actually determines what the structure will all be. So whenever you have a protein, if you know the tertiary structure or the 3D structure, you'll be able to determine how ligand will bind with any specific protein and at what point the exact angle it is going to bind. So the fourth level, which is the quaternary, that is when you have multiple chains forming just one complex. And as you have it on the screen, you can easily see in terms of the primary structure, you know the difference when it comes to the secondary structure of a protein, which can either be anticlockwise, or it can be clockwise. If it is anticlockwise, that means that protein is going to contain beta sheet. So then the tertiary one, when you combine two secondaries together, that's going to form tertiary. And the quaternary, just from the word quati, that means that is a quarter. So that means when you have four protein complexes coming together, will give you quaternary. Okay. Now, what are our main targets of today? I told you we are going to make use of 4 different bioinformatics tools to be able to study the structure of protein and their function. The first one is UniProt. UniProt is just a database at which we can successfully retrieve information about protein, and that is the sequences and their functional data. Then the second one, which has to do with the structural database, which is called PDB. If you are looking for the sequences, of any protein, you go to UniProt. Then if you want to know their structure, their 3-dimensional structure, that is when you go to PDB, and that is very, very important for you. The next one is when you see when we will still make use of Expasy, which has to do with analyzing protein properties by using Expasy as a tool. We are going to make use of that. Then PyMOL. PyMOL is a visualizing protein database when it comes to using that. Okay. Now, a demo of UniProt usage. The first thing is you type in www.uniprot.org, and we'll look for an enzyme which is called beta galactosidase with the accession number, which is known as a P0722. And by the time you do that, that is what is going to display to you. You are going to see the name of that enzyme, that is the gene. You see the gene name, you see the Escherichia coli, which is this, and you see the number. So at that point in time, you will be able to know the function, whether it is a subcellular localization, wherever that is, the point at which it is located, and so on and so forth. Don't forget that most of the format we normally use in bioinformatics is a faster. So do that. So save faster format, or you can screenshot it for us that are using smartphones. Okay. Now, demo for how to use PDB. That is, if you want to retrieve information that has to do with knowing the structure of that protein. So you go to rsb.org, then you type in the name, which is a beta galactosidase, with the PDB identity, which is called CDRV. And as you can see it on the screen, beside the text, you will see the image of that. So at that point in time, you'll be able to know how many chains. How many chains? Is it going to be alpha chains, beta chain, or what? So you will see it there. Then after that, you will download that file for you to be able to see it later on. Then you can also check the resolution, the method by which they use in getting that protein, and you will be able to know how many amino acids. Like that of beta galactosidase, it is between amino acid 1 to 581. So you need to document all this for you to be able to visualize the protein structure from the protein database, which is PDB. Now going to the demo for using Expasy tools. So the first thing you do is to type expasy.org, and when you type expasy.org, you are going to make use of what is called protoparam, for you to be able to see the molecular weights, the protein index, you'll be able to see stability. All these properties of protein will go a long way to determine the functions. We go a long way to see, for example, those of us that might want to use some proteins for vaccine design. So all these are very, very important. Even in drug design, your protein candidate has to be very, very stable. It has to have a very appropriate molecular weight, as well as the protein index. It has to be there. Then not only that, at this expasy.org, you'll be able to determine the hydrophobicity. That is, what are the properties? I mean, how can in terms of water hating and water loving properties, what are those ones that that can actually give you at that level? So that is going to give you that. And if you look at what you have on the screen, you will see that this is the amino acid. The molecular weight is more than 27 million points or so Dalton. Then you see that the theoretical PI is around 6.79. So all these properties will go a long way to determine the functionality of your protein. Demo for visualizing structures in PyMOL. So that is for us that are using laptop or system. So you have to download the PDB file in PyMOL. The PDB that you downloaded the other time, it is now time for you to be able to upload it there. By the time you upload it, you'll be able to zoom, you rotate. Sometimes you also apply different color to differentiate the active sites, the ligand, and the protein. You're able to do that. Then it is also very possible for you, when you are using PyMOL, to be able to measure distances, and you'll be able to save the image as it is. So because there is if there's going to be a rotational index whereby you will see it in that. And if you look at what you have on the screen, you will know definitely very well that the angle of binding, binding angle is very, very important in going a long way to determine the properties of any protein, so to say. And when the time comes, there's going to be hands-on exercises on how can you use UniProt for you to be able to determine the function of RNA polymerase. By this time around, by now, you should know the function of RNA polymerase. That is exactly what the enzyme that starts transcription, okay? And you should be able to retrieve structure from PDB, and you'll be able to take note of some features. Then you'll be able to run protoparam on Expasy using basic statistics. Then you should be able to visualize your structure in PyMOL. All this should be done even offline whenever you are even that. Then there are other offline motifs. You should be able to know PDB format. That is how you are going to see it. That's going to be how it is going to appear to you. Whenever you have faster format, it is going to be different from that, and you use notepad to be able to have that. Then screenshotting some of these structures might also go a long way to be able to do that. Then you should be able to learn how to do your documentation ready without the use of without the use of internet. Okay, so a time you should be able to apply, you should be able to attempt all these quizzes. For example, what is typically found in UniProt entry? What are the levels, 4 levels of a protein structure? You should be able to know which database is used for 3D structures. What are the tools for computing PI of any protein, and what do you understand? And why, what is PyMOL being used for? So, without wasting our time, you should be able to, the next thing, you should be able to go for the classwork, which is going to be protein structure and function determination. And the objective of this is to be able to use tools like UniProt, PDB, Expasy,